Hello students of class 12, welcome back once again to your biology class. Today we will start a new lesson. We will know about the chapter of microbes in human welfare. You all know that microbes are the organisms which cannot be seen by our naked eyes. And where are the microbes found? These are found everywhere in the soil, water, air and inside even the bodies of living organisms. And these microbes can also be found in thermal vent, deep inside the soil, under snow, as well as the acidic environment also. Microbes can be of various varieties. They can be protozoa, bacteria, fungi, plant virus, viroids, and even prions. So various microbes can be found in the environment. You can see the phage virus, that is bacteriophage, adenovirus, tobacco mosaic virus. These are the various types of microbes which can be found. And the colonies of the bacteria, they grow, and fungal colonies also they are found. Now we will know about the microbes in the household products. At your home, what microbes or how microbes are used? First, curd. You know curd, how it is formed? Curd is formed by adding a small amount of curd to milk. At your home, you have seen your mother making curd from previous curd by adding small quantity of milk. Now here the lactobacillus or the lactic acid bacteria, in short we say LAB, which is present in the starter, they multiply when they get a suitable environment and they convert milk into curd. The acids which are released by the lactobacillus during the growth, what they do, they coagulates and they partially digest the milk, right? And um, the protein part of the milk is basically digested. The name of the protein part of the milk is casein. Now what are the benefits of this LAB or lactobacillus, lactic acid bacteria? They increase the vitamin B12 amount and so increase the nutrient quality of the milk, and these also check the disease causing microbes in the stomach. Next, how microbes are used in household products. Dough, how it is formed? By the fermentation of bacteria. Now bread, how it is made? By fermentation of Saccharomyces cerevisiae, or commonly we say as baker's yeast. And the carbon dioxide which is released during the process of fermentation, they give the puffy appearance to the dough. And this also is used for making different type of food items like idli, dosa, bread, etc. Okay, so in this manner the puffy appearance is made. Now toddy, you know, it is a national or traditional drink, we can say it's a traditional drink of southern India. The people of southern India take toddy, and this toddy, how it is made? It is made by fermentation of sap from the palm trees by using different types of bacteria. And cheese is also formed by partial degradation of milk by different microorganisms. Swiss cheese, you have heard about the name. It is formed by the bacterium Propionibacterium shermani, and its characteristic feature is, what is the characteristic feature of Swiss cheese? I think you know, you have seen in the cartoon Tom and Jerry. Its characteristic feature is the formation of large holes within the cheese. Why these holes are produced? Due to the production of large amount of carbon dioxide. Okay, another is the Roquefort cheese. Roquefort cheese, it is formed by ripening with the fungi Penicillium roqueforti for a particular flavor. So this is flavored cheese. Now we will know about microbes in industrial products. Large scale production of various compounds in industries is done in a very large vessel known as fermenters. Okay, and these fermented beverages, how these are formed? These are formed by fermenting the fermenting the malted cereals and the fruit juices with Saccharomyces cerevisiae, or we say brewer's yeast, to produce the ethanol. And the formation of different types of alcoholic drinks, it depends on the raw material and the type of processing. Wine and beer are produced without performing the process of distillation, and whiskey, brandy, and rum, these are formed by the process of distillation. And antibiotic production is also another usage. As industrial product, antibiotics are also used. What are antibiotics? Antibiotics are the chemical substances which is produced by some microorganisms which can kill or retard the growth of other disease causing microorganisms. Penicillin, it was discovered by Alexander Fleming, and it was the first antibiotic which was discovered. While working with Staphylococcus bacteria, Fleming observed that the growth of the mold around the bacteria did not grow. Okay, what he observed, that the growth of the mold which was growing around which the bacteria did not grow, and it was found to be a chemical penicillin which was produced by Penicillium notatum. In this way, this penicillin was discovered by Alexander Fleming. And this disease cured by antibiotics can, the many diseases can be cured by using these antibiotics, like plague, whooping cough, which is known as the kali khasi in local languages. Diphtheria and leprosy also can be cured by using these antibiotics. Next is production of different types of organic acid, alcohol, and enzymes. Now, different organic acid are produced by microbial metabolic actions. Okay. Now, citric acid, acetic acid, butyric acid, lactic acid. These all are type of microbes produced. Citric acid is formed from fungi, and acetic acid, butyric acid, and lactic acid, for this formation, a special type of bacteria is used. Okay. Now, citric acid, what is the microbe used for the production of citric acid? Aspergillus niger. Acetic acid is produced from the bacteria Acetobacter aceti. Butyric acid is produced from the bacteria Clostridium butylicum, and lactic acid is produced by using the microbe Lactobacillus. Okay, so these are the organic acid which are produced by using different types of microbes. Apart from that, ethanol is also produced by Saccharomyces cerevisiae, which is yeast. Clear? And enzymes are commercially also produced. Different types of enzymes, lipase, pectinase, protease, streptokinase. These are also produced artificially. And what are the uses of lipase? You have to know. Lipase is used in detergent formulations, and it helps removing the oily stains from different types of um dresses. Oily stains, if it comes on dresses, it helps in removing. So it is used in detergent formulation. What? Lipase. And pectinase and protease is used for clarifying the bottle juices. And streptokinase is produced by streptococcus and modified by genetic engineering, and the streptokinase is used as a clot plaster for removing the clot from the blood vessel of the patients who have undergone myocardial infarction. Okay, clot blaster remover. And different types of bioactive molecules are also produced by using these microbes, like cyclosporin A. What is the source organism? This is cyclosporin A is produced from a fungus. What is the name of the fungus? Trichoderma polysporum. Okay, and what is the use? The cyclosporin A is used as immunosuppressive agent in organ transplant patients. Those who are having organ transplantation, their cyclosporin A is used as immunosuppressive agent in those patients. Another molecule is statin. Statin is a bioactive molecule which is produced from a fungus, Monascus purpureus. And where is this statin used? This is used as blood cholesterol lowering agent. Okay, and it completely inhibits the enzymes for synthesis of cholesterol. So statin is used in these cases. Now we will know about microbes in sewage treatment. Sewage, you know what is a sewage? Sewage is the municipal wastewater which contains large quantities of human excreta. Now, what is the need for sewage treatment? Sewage, what it contains? It contains large amount of organic matter and pathogenic microbes. And before discharging into the natural bodies, this sewage is made less polluting. As a result, some treatment should be given to the sewage before it is released to the water bodies. And where is sewage treated? The sewage is treated in sewage treatment plant, or we say STP, sewage treatment plant. Now, for treating this sewage water, two plans has been initiated by the Ministry of Environment and Forest. One is Ganga Action Plan, another is the Yamuna Action Plan. Okay, and here large number of STP. What I said, STP is the sewage treatment plant. It will be built up to treat the sewage before they are discharged into the Ganga and the Yamuna rivers to reduce their pollution. Now this sewage treatment is carried out in two stages. First is the primary treatment or physical treatment. Second is the secondary treatment or biological treatment. Now primary treatment, what is that? It is the physical removal of particles. It is the physical removal of large and small particles from sewage. First, what happens here? The floating debris or the light debris is removed by sequential filtration by passing through wire mesh screens. Okay, then what happens? The grit. Grit means the soil and the small pebbles. These are removed by sedimentation in the settling tank. And the sediment is called the primary sludge, and the supernatant, what is left is the effluent. And then this effluent is taken for secondary treatment. In the secondary treatment or biological treatment, this primary effluent is passed into large aeration tanks with constant mechanical agitation and air supply. Useful aerobic microbes, they grow rapidly in this situation and they form flocks. Now, what are these flocks? Flocks are the masses of bacteria which is associated with fungal filaments to form mesh-like structures. And the growing microbes, they go on consuming the organic matter, thereby reducing the biochemical oxygen demand, which is known as the BOD. When the BOD or biological oxygen demand of sewage has reduced, then this effluent is passed into the settling tank. Here what happens? The bacterial flocks settle and the sediment is called activated sludge. A small part of this activated sludge is used as an inoculum in the aeration tank, and the remaining part is passed into large tank called anaerobic sludge digesters. In the digesters, what happens now? The heterotrophic microbes anaerobically digest the bacteria and the fungi in the sludge and produce the mixture of gases like methane, hydrogen sulfide, and CO2. And these basically forms the biogas. Okay, now this biological oxygen demand, I said BOD, it refers to the amount of oxygen which is consumed if all the organic matter in 1 liter of water is oxidized by bacteria. BOD means measures. Biological oxygen demand measures the amount of the organic matter in water by measuring the rate of oxygen uptake by the microbes. Higher biological oxygen demand will indicate higher polluting environment. Okay, so you can understand that if BOD is more, then higher polluting potential we can understand from there. Now you can see here it is given in tabulated form. The primary effluent aeration tank, it is going to it's happening constant agitation. After that, the aerobic bacteria influx consume the organic matter and biological oxygen demand reduces. And in case effluent to the settling tank directly goes to natural water bodies, or else it goes to flux where it sediment and forms the activated sludge, and this anaerobic sludge digester, through this anaerobic sludge digester the anaerobic bacteria digest the sludge here and release different types of gases which is known as the biogas. Okay, here you have to okay, leave it then. You have to make out this treatments, the sewage treatment. Here it is given, you can see I said this, the Ganga action plan and the Yamuna action plan. Now we will know about microbes in the production of biogas. Now biogas, what is that? It is a mixture of inflammable gases like methane, sulfur dioxide, carbon dioxide, which is produced by the microbial activity that can be used as fuel. You know, biogas nowadays has become very common and it is used for cooking purposes in different parts of India. Methanogens, particularly the methanobacterium, what they do, they anaerobically break down the cellulosic material and forms CO2 and H2 in anaerobic sludge in sewage treatment plant and also in the rumen of the cattle. What is rumen? I think you know rumen is a part of the stomach of the cattle and they provide nutrition to the cattle. Okay, and these Indian Agricultural Research Institute and Khadi and Village Industries Commission, they developed the technology of biogas in India. Now we will know about the parts and the mechanism of biogas or gobar gas. Okay. Now the raw material for biogas production is the excreta or the dung of the cattle. We all know from this excreta or dung of the cattle the biogas is produced. What here happens is in the biogas plant, you have to draw and level the structure of a biogas plant. The biogas plant has a concrete tank, it's about 10 to 15 feet deep, in which the bio waste and the slurry of the dung is collected first. The tank, it has a floating cover which rises on the production of the gas in the tank. Understood? Whenever the gas is produced in the tank, the floating cover of the tank rises up due to pressure. Methanobacterium, which is present in the dung, may act as bio-waste and produce the biogas, and the gas produced that is supplied to the nearby houses by an outlet. And through another outlet, the spent slurry is removed and this can be used as fertilizer. This biogas, what it is used for? As fuel for cooking and lighting also. Now microbes as biocontrol agents. Biocontrol, biocontrol is defined as the controlling of the plant diseases and pests by using the biological methods. And earlier, different types of chemicals like insecticides and pesticides were used for biocontrolling. Nowadays, different types of organic usage has been motivated. Now, these chemical agents have some disadvantages. These chemicals are very toxic and harmful to the human beings and the animals, and they also pollute the environment and the plants. And the weedicides, which are used to remove the weeds, they also pollute the soil. So, as a result, there is a holistic approach for organic farming, which keep the pests at manageable level by a complex system of checks and balances within the ecosystem instead of their complete eradication. Now, there are some approaches for biological farming. What is that? First, familiarity should be there with various life forms inhabiting the field. And apart from that, the person who is going for organic farming, he should have the knowledge about the life cycle patterns of feeding and the habitat of predators and the pest. Ladybugs, dragonflies, they are used to get rid of aphids and mosquitoes. BT, that is Bacillus thuringiensis, that is used to control the butterfly caterpillars. What happens here? The dried spores of BT are mixed with water and those are sprayed on plants like brassicas and fruit trees. Insect larvae, after eating these, they are killed by the toxin which is released in their gut. Bacillus thuringiensis toxin gene, they have been introduced into the plants to provide resistance to the pest like BT cotton we say. And trichoderma also, the free living fungi, these are present in the root ecosystem where they act against several plant pathogens. Apart from that, baculovirus is also there. They are the pathogens which attack the insect and other arthropods. Okay, baculoviruses are very helpful in integrated pest management, we say in short IPM program, in which the beneficial insects are conserved and they don't harm the plants, mammals, birds, fish and other non-target insects also. Clear? Now we will know about microbes as biofertilizer. Biofertilizer, what are these? These are the microorganisms which enrich the nutrient quality of the soil. Nutrient quality means nitrogen, phosphorus, potassium. Bacteria, fungi and cyanobacteria are the three main sources of biofertilizers, Rhizobium, Azospirillum, then Azotobacter. These are the bacteria which are used as biofertilizers. Rhizobium, it has symbiotic association. It is a symbiotic bacterium which lives in the root nodules of leguminous plants, and what they do, they fix the atmospheric nitrogen into organic compound. Another is Azospirillum and Azotobacter. What they do, they are also free living bacteria and they absorb the free nitrogen from the soil, air, and they convert it into salts of nitrogen like amino acid and enrich the soil nutrients. As a result, the plant can get nutrients from the soil. Okay, next, fungi as biofertilizer. Fungi can also be used as biofertilizers. What they do, they form the symbiotic association with the roots of higher plants called mycorrhiza, like glomus, we can say. And the fungal hypha, what they do, they absorb the phosphorus from the soil and they pass it to the plant. Mycorrhiza, they show different benefits. What are the benefits? Like they show resistance to root-borne pathogen. They can tolerate to salinity and drought, and the overall increase in the plant growth and the development is also taken place. Okay, and cyanobacteria or blue-green algae, they also act as biofertilizer. What they do, they fix the atmospheric nitrogen and increase the organic matter of the soil through their photosynthetic activity. You can give some example of Anabaena nostoc oscillatoria. And these blue-green algae or cyanobacteria, they increase the fertility of the soil and add the organic matter to the soil. Okay, so in this way, different types of algae, fungi and cyanobacteria act as biofertilizers. So this is all about the chapter you have to learn. So till this much today. Thank you.